Before we dive in, it's crucial to state that the information in this presentation is for educational and informational purposes only. It is not a substitute for professional medical advice, diagnosis, or treatment. Always seek the advice of your physician or other qualified health provider with any questions you may have regarding a medical condition. Never disregard professional medical advice or delay in seeking it because of something you have seen in this video. Let's be honest. For decades, the conversation around weight loss has been dominated by two words: eat less, move more. It's a simple formula, a mantra repeated by doctors, trainers, and well-meaning relatives. And for millions of people, it's a recipe for failure. You've been there. You've counted the calories, you've sweated on the treadmill, you've sworn off carbs, sugar, and everything delicious. You lose 5 pounds, 10 pounds, maybe even 20. You feel a glimmer of hope. And then life happens. The weight creeps back on, often bringing a few extra pounds with it. Each time it feels like a personal failure, a lack of willpower, a character flaw. But what if it's not? What if, for many, this isn't a battle of willpower, but a battle against your own biology? What if your body has a complex, ancient system designed to hold on to weight, a system that fights you every single step of the way? And what if we've finally found a way to speak to that system in its own language? Suddenly, everyone is talking about it. Ozempic, Wegovy, Mounjaro. These names are everywhere, from celebrity gossip columns to the cover of Time magazine. They are being hailed as revolutionary, miraculous, a game changer in the fight against obesity. We're seeing weight loss results that were previously only possible with invasive surgery. People are quieting the constant food noise in their heads for the first time in their lives. The demand is so high it's causing worldwide shortages. But behind all the hype, all the headlines, and all the controversy, lies a simple, critical question that most people can't answer. How? How do these injections actually work? What is happening inside your body after that pin clicks? It's not magic, it's science. And understanding that science is the key to understanding the future of weight loss. Over the course of this deep dive, we are going to pull back the curtain. We're not just going to scratch the surface, we are going to go deep into the intricate biological dance that makes these medications possible. By the end, you will understand the science behind GLP-1 agonists better than 99% of the population. You'll know how they command your brain, slow down your stomach, and fundamentally rewire your relationship with food. Whether you're considering these medications, are already taking them, or are just fascinated by the biggest medical story of our time, this knowledge will empower you to have more informed conversations with your doctor and to truly grasp what's happening inside your own body. Our story doesn't start in a modern pharma lab. It starts almost a century ago, with a puzzling observation that stumped scientists for decades. It was called the incretin effect. Here's the puzzle. Doctors knew that when blood sugar rises, the pancreas releases insulin to manage it. Insulin is like a key. It unlocks your cells to let the sugar in for energy. Simple enough. So they did an experiment. They took a group of healthy volunteers and gave them a dose of sugar glucose directly into their bloodstream within 4. They measured the insulin response. It was predictable. Then, they took the same group of people and had them drink the exact same amount of sugar. Logically, the insulin response should have been the same, right? Same amount of sugar, same result. But it wasn't. It wasn't even close. When people drank the sugar, their pancreas went into overdrive, pumping out a massive wave of insulin far, far more than when it was injected. Think about that. The simple act of the sugar passing through the digestive system, the gut, was triggering a completely different supercharged response. The gut was clearly sending a powerful signal to the pancreas, a message that said, "Get ready. A big load of fuel is coming down the pipe." For years, this mysterious signal was just a theory, a ghost in the machine. Scientists called these hypothetical messengers incretins, a name that comes from intestine, secretion, and insulin. They knew these incretins existed, but they couldn't find them. It was a biological manhunt. And then, in the 1980s, they finally captured the culprits. They found not one, but 2 key hormones responsible for this effect. One was called GIP, and the other, the hero of our story, was called Glucagon-like Peptide-1, or for short GLP-1. This was the breakthrough. GLP-1 was the messenger. When you eat a meal, specialized cells in your small intestine called L cells release GLP-1 into your bloodstream. It then travels through your body like a biological text message, delivering critical instructions to different organs. Its most famous job, the one that got scientists so excited, was talking to the pancreas. It told the pancreas to release insulin, but it did so in a remarkably intelligent way. It was glucose-dependent. This means GLP-1 only tells the pancreas to make insulin when blood sugar is high. If your blood sugar is normal or low, GLP-1 doesn't do much. This is a brilliant safety feature built by nature. It prevents your blood sugar from crashing, a dangerous condition called hypoglycemia. So, right away, researchers saw the potential. Here was a natural hormone that could help people with type 2 diabetes control their blood sugar in a smart, safe way. But as they studied GLP-1 more, they realized it wasn't just a one-trick pony. It was sending messages all over the body. They discovered it also talks to the stomach. It sends a signal that slows down gastric emptying. In simple terms, it makes the food you've eaten sit in your stomach for longer. This has a powerful effect. It makes you feel physically full for longer after a meal. That feeling of being stuffed after Thanksgiving dinner? GLP-1 helps create that. And then came the biggest discovery of all. They found that GLP-1 could cross the blood-brain barrier. It was traveling all the way up to the command center of the body, the brain, and directly influencing the hypothalamus, the area that controls appetite and satiety. It was the biological equivalent of your gut picking up a direct phone line to your brain and saying, "Hey, we're good down here. We've got plenty of energy. You can stop feeling hungry now. Put the fork down." This was revolutionary. The eat less part of the weight loss equation wasn't just about willpower anymore. Here was a hormone that physically reduced hunger at its source. It turned down the volume on the food noise that so many people struggle with. So now the picture was complete. GLP-1 was a powerhouse. It controlled blood sugar, it made you feel physically full in your stomach, and it told your brain you weren't hungry anymore. It was nature's perfect appetite suppressant and blood sugar regulator, all rolled into one. There was just one massive problem. A problem that almost made the entire discovery a scientific dead end. Natural human GLP-1 has a half-life of less than 2 minutes. Let me say that again. 2 minutes. From the moment your gut releases this incredible hormone, your body produces another enzyme, called DPP4, that immediately hunts it down and shreds it to pieces. It's designed to be a short, sharp signal, not a lingering presence. So, you eat, you get a quick burst of GLP-1, the signal is sent, and then poof, it's gone. This biological reality presented an impossible challenge for drug development. How could you possibly turn a hormone that vanishes in 120 seconds into a viable treatment? You couldn't have patients hooked up to a constant 4-drip of GLP-1 for the rest of their lives. The dream of harnessing this powerful pathway seemed to be over before it even began. Scientists needed to find a way to make the signal last. They needed a version of GLP-1 that could survive the brutal environment of the human bloodstream for hours or even days. They needed a molecular superhero. And the clue to creating one wouldn't be found in a high-tech lab, but in the saliva of a venomous lizard, hiding under a rock in the deserts of the American Southwest. The year is 1990. Our story moves from the gleaming boardrooms of pharmaceutical companies to a much humbler setting, a Veterans Affairs hospital in the Bronx. Here, a brilliant but unassuming endocrinologist named Dr. John Ung was on a personal quest. He wasn't hunting for a blockbuster drug. He was driven by pure scientific curiosity. He was fascinated by how the gut and the pancreas talk to each other, and he spent his days screening the venom of various creatures, looking for new hormones that might regulate this conversation. It was a strange hobby, almost an obsession. He believed that nature, in its infinite and sometimes brutal wisdom, had already solved many of the problems that plagued human biology. He was a molecular treasure hunter, and he was convinced the map was written in the DNA of the world's most extreme survivors. One day, he came across a paper describing the Gila monster. This wasn't just any lizard. It was a creature of incredible extremes. Living in the harsh deserts of Arizona and Mexico, the Gila monster is a master of energy conservation. It might only eat 4 or 5 massive meals a year. Imagine that. It will gorge itself, consuming up to a third of its body weight in a single sitting, and then fast for months. For any other animal, a sudden flood of nutrients and sugar like that would be catastrophic. It would send its blood glucose into a dangerous organ-damaging spiral. The pancreas would be forced to pump out so much insulin it would essentially burn itself out. This process sounds eerily familiar, doesn't it? It's a sped-up version of what happens over years to a person developing type 2 diabetes. But the Gila monster was fine. Perfectly fine. Dr. Ung had a flash of insight. This lizard must have evolved an incredibly powerful, incredibly efficient system to manage that sugar spike. Its body had to produce a signal that told the pancreas, Get ready, a massive amount of food is coming. Start producing insulin now, but do it in a controlled, steady way. Don't panic. He suspected that signal would be a hormone very similar to human GLP-1. But it would have to be different. It would have to be stronger, more robust. It would need to stick around for a long time to manage a meal that was being digested over days, not hours. This was his X marks the spot. He believed the Gila monster's venom, a complex cocktail of proteins and peptides, held the key. So he did what any curious scientist would do. He picked up the phone. He found a venom supplier in Utah, a man who wrangled these creatures for a living, and he ordered a sample. When the vial of venom arrived at his lab in the Bronx, it was like receiving a message from 40 million years of evolution. He began the painstaking work of separating the venom into its individual components. Using a technique called chromatography, he meticulously isolated molecule after molecule, testing each one to see what it did. And then he found it. A peptide he had never seen before. He named it Exendin-4. When he sequenced Exendin-4, his jaw must have hit the floor. Its molecular structure was a stunning 53% identical to human GLP-1. It was like finding a long-lost cousin. It was clearly designed to do the same job, to bind to the GLP-1 receptors in the pancreas and signal insulin release. It was nature's version of the exact hormone that scientists were struggling to turn into a viable drug. But here's where it gets truly incredible. Remember that villain from our story, the enzyme DPP-4? The molecular scissors that snip GLP-1 in half in less than 2 minutes? Dr. Ng tested his new lizard peptide against it. He mixed Exendin-4 with the DPP-4 enzyme and waited. And waited. Nothing happened. The DPP-4 enzyme couldn't touch it. It was completely immune. While human GLP-1 was a fragile mayfly, living for a fleeting moment, Exendin-4 was a biological battleship. It could circulate in the bloodstream not for 2 minutes but for over 12 hours. This was the holy grail. The molecular superhero they had been looking for. A version of GLP-1 that could survive the journey through the human body long enough to actually do its job. So what was the Gila monster's secret? What was the molecular trick that made its hormone so invincible? The answer is a masterclass in the power of evolution. It came down to a single tiny change in its chemical blueprint. Imagine GLP-1 is a key, made up of a chain of building blocks called amino acids. The DPP-4 enzyme is a very specific lockpicker. It recognizes the first 2 amino acids on that chain, latches on, and breaks the key. The second amino acid in human GLP-1 is a molecule called alanine. This is the weak spot. This is the target that DPP-4 always attacks. But in the Gila monster's Exendin-4, evolution had made a substitution. In that critical second position, there wasn't an alanine. There was a different amino acid called glycine. This one tiny change was everything. It was like putting a different shaped head on the key. The DPP-4 enzyme, our lockpicker, came along, tried to grab it, and its tools just slipped off. It couldn't get a grip. By changing just one single amino acid out of 39, the Gila monster had created a version of GLP-1 that was effectively invisible to its natural predator. It was a perfect molecular disguise. This is why these new medications are called GLP-1 receptor agonists. They aren't actually GLP-1. They are mimics, imposters, biological actors that play the part of GLP-1 so perfectly that our bodies are fooled. But they play the part for much longer. Dr. Ng, working in his humble VA lab, had uncovered a secret that had eluded massive pharmaceutical companies for years. He patented his discovery in 1993. He had found the blueprint for a revolutionary new class of diabetes drugs. The story should have ended there, with him being hailed as a hero, right? But science is rarely that simple. A discovery is one thing. Turning it into a medicine that can be prescribed by a doctor is another. Dr. Ng's groundbreaking patent, filed from a hospital in the Bronx, sat there. It was a brilliant piece of science, but it was just a piece of paper. The world didn't know it yet. But the key to a multi-hundred-billion-dollar medical revolution was collecting dust in a filing cabinet. Because to take this lizard venom molecule and get it into a pharmacy, it would take a different kind of hero. It would take someone with the vision, the resources, and the sheer audacity to bet millions of dollars on a hunch that a substance designed to incapacitate prey in the desert could be re-engineered to safely treat a chronic disease in millions of humans. The science was solved, but the business was just beginning. And the company that would ultimately take that bet wasn't a giant like Merck or Novo Nordisk. It was a tiny, struggling biotech startup that was on the verge of going bankrupt. That company was Amylin Pharmaceuticals. And in the late 1990s, Amylin was in a death spiral. Based in San Diego, they had poured everything they had into a different diabetes drug, and it had failed. Miserably. Their stock had cratered, investors were furious, and the vultures were circling. They were burning through cash with nothing to show for it. To the outside world, they looked like just another biotech flameout. A cautionary tale whispered in the halls of venture capital firms. But inside Amylin, a small group of scientists saw a flicker of hope in the most unlikely of places. The work of a lone researcher in a Bronx VA hospital, and the venom of a desert lizard. They had read Dr. John Ng's papers. They understood the monumental breakthrough he had made. While the rest of the pharmaceutical world was trying to solve the GLP-1 puzzle by tweaking the human molecule, Ng had found a ready-made solution in nature. A key that not only fit the lock but refused to break. This molecule, exendin-4, was a gift from 30 million years of evolution. It was nature's brute force solution to a complex biological problem. And Amylin realized this wasn't just a scientific curiosity. This was a lifeline. It was a bet the company kind of decision. A Hail Mary pass in the final seconds of the game. Imagine the board meeting. The CEO trying to convince a room full of skeptical, money-conscious investors. So let me get this straight. You can almost hear them saying, "Our first drug failed. We're almost out of money. And your brilliant new plan is to develop a drug based on lizard spit." Sounds insane. It sounds like something out of a comic book. But desperation is a powerful motivator. With nothing left to lose, Amylin decided to go all in on the Gila monster. They licensed the patent for exendin-4 from Dr. Ng, but that was the easy part. Now came the hard part. The impossibly hard part. How do you turn a component of raw venom into a safe, reliable, mass-produced medicine for millions of people? The first problem was the source. You can't just build a giant Gila monster farm in the California desert and go around milking them for their venom every day. These are not cows. They are reclusive, protected, and frankly very grumpy reptiles that only produce minuscule amounts of venom. The logistics were a nightmare, the cost would be astronomical, and the supply would be ridiculously unstable. The entire idea was a non-starter. If this was going to work, they couldn't rely on the lizard. They had to bypass it entirely. They had to build the molecule themselves from scratch in a lab. This is the miracle of modern biotechnology. Peptides like exendin-4 are simply long chains of amino acids, the basic building blocks of protein. Think of them like a very specific pearl necklace. If you know the exact sequence and type of pearls, you can, in theory, string them together to make an identical copy. Amylin's chemists had the sequence, thanks to Dr. Ng's research. Their task was to become master jewelers on a microscopic scale. Using a process called solid-phase peptide synthesis, they began the painstaking work of linking one amino acid to the next, in the exact order dictated by the Gila monster's DNA. It's an incredibly precise and delicate process. One wrong amino acid, one broken link, and the entire chain is useless. The molecule won't fold correctly, it won't fit the GLP-1 receptor, and the drug will fail. For months, their labs worked to perfect the recipe, creating a synthetic, man-made version of exendin-4. A molecule that was chemically identical to the one found in the lizard's venom, but born in the sterile steel vats of a biotech facility. They called their new drug exenatide. With a pure synthetic supply of the drug secured, they could finally move on to the next terrifying hurdle: testing it in human beings. This is the moment of truth for any new drug. You can have the most brilliant theory, the most elegant chemistry, but until you inject it into a person, you have no idea what will happen. The human body is infinitely complex. Will it be safe? Will it be toxic? Will it trigger a massive immune reaction? Or will it, just maybe, actually work? The first human trials, known as Phase 1 trials, are all about safety. They started with a small group of healthy volunteers. Imagine being one of those first people. You're sitting in a clinical trial facility, and a doctor explains they are about to inject you with a synthetic chemical originally discovered in the venom of a poisonous lizard. You'd have to be pretty brave. The researchers at Amylin held their breath. They administered the first tiny doses. And they watched. They monitored vital signs, they took blood samples, they waited. And nothing catastrophic happened. There were no immediate, dangerous side effects. The drug appeared to be safe. That was the first massive sigh of relief. But then came the next part. They gave the drug to people with type 2 diabetes. And what they saw was astounding. It was everything Dr. Ng had predicted and more. When patients ate a meal, their blood sugar didn't spike wildly like it normally would. The synthetic lizard molecule was commanding their pancreas to release the perfect amount of insulin at the perfect time. It was working. It was actually working. The data coming back from the labs was almost too good to be true. The Hail Mary pass looked like it might actually be caught. But they also noticed something else. Something they hadn't fully expected. The patients in the trial started reporting a curious side effect. They weren't as hungry. They felt full faster. At mealtimes, they'd put down their forks halfway through a meal they would have normally finished. They were losing a little bit of weight. It wasn't the primary goal of the study, that was blood sugar control, but it was an undeniable, consistent observation. This was the first clinical proof of what scientists had suspected in the lab. This GLP-1 pathway wasn't just about insulin and blood sugar. It was a master regulator of the entire metabolic system. It was talking to the pancreas, yes, but it was also sending powerful signals straight to the brain telling it, hey, we're good. We've got enough fuel. You can stop eating now. For the team at Amylin, this was a revelation. They hadn't just created a new diabetes drug. They had stumbled upon a key that unlocked the very biology of appetite and weight. The potential was staggering, far beyond what they had initially dared to dream. The science was sound, the drug was working, and it was safe. They had climbed a mountain. They had taken a wild gamble on lizard venom, and it was paying off in spectacular fashion. They had a potential blockbuster drug on their hands, a drug that could change the lives of millions and transform the treatment of diabetes. But as they stood on that mountaintop, celebrating their incredible achievement, they looked ahead and saw an even bigger mountain looming in the distance. A mountain they had no possible way of climbing alone. Because Amylin was still a tiny company. They had the science, they had the patent, they had the early clinical data. But they didn't have the hundreds of millions or even billions of dollars it would take to fund massive global phase 3 trials, navigate the labyrinthine FDA approval process, build a global manufacturing and supply chain, and hire a sales force to market the drug to doctors around the world. They had the miracle molecule, but they didn't have the muscle to bring it to the world. To do that, they needed a partner, a giant. They needed to go back to the very same big pharma companies that had overlooked the science in the first place, and convince one of them to take the biggest gamble of all. The pitch meetings must have been a sight to see. On one side of the polished mahogany table, you have the titans of the pharmaceutical industry, the executives from Merck, Pfizer, Johnson & Johnson, men in $1,000 suits who controlled budgets larger than the GDP of small countries. And on the other side, you have the scientists from Amylin Pharmaceuticals. Passionate, brilliant, but in the grand scheme of things, a tiny company from San Diego. They walk into these boardrooms, these temples of modern medicine, holding not much more than a collection of scientific papers and a story that sounds like it was ripped from a comic book. Imagine their presentation. Good morning, gentlemen. We're here today to ask for $1 billion investment to develop our next blockbuster drug. It's a revolutionary new treatment for diabetes. And we discovered it in the venom of a Gila monster. You can almost hear the crickets, the polite but deeply skeptical smiles, the barely suppressed chuckles. Lizard spit? You want us to bet the company on lizard spit? In an industry built on precision chemistry and sterile labs, this was biological alchemy. It was messy. It was weird. And for most of the big players, it was a hard pass. One by one, the giants of pharma turned them down. The idea was too strange, the source too bizarre, the risk too high. It seemed that the Gila monster secret was destined to remain just that. A fascinating but commercially unviable footnote in the annals of science. But Amylin kept knocking on doors. Because they knew what they had. They knew this wasn't just a curiosity. It was a fundamental breakthrough in understanding metabolism. All they needed was one company, just one, with the vision to see past the lizard and focus on the science. And finally, they found it. In Indianapolis, Indiana. The home of Eli Lilly and Company. Now, Eli Lilly was no stranger to diabetes. They were the first company to commercialize insulin back in the 1920s. Diabetes was in their corporate DNA. But by the early 2000s, their pipeline was looking a little dry. They needed a win. They needed something new, something that could redefine the market. And when the team from Amylin came to them with the story of Exendin 4, someone at Lilly listened. Someone saw the potential. They looked at the data, the blood sugar control, the way it protected beta cells, the tantalizing hints of weight loss, and they understood. This wasn't just another Me Too drug. This was a whole new class of medicine. This was a paradigm shift. So, in 2002, Eli Lilly did what no one else would. They took the gamble. They signed a massive deal with Amylin. A partnership that would pour hundreds of millions of dollars into developing this strange molecule from the desert. The race was officially on. But getting the deal signed was just the first hurdle. Now came the monumental task of turning a peptide found in lizard venom into a safe, effective, and convenient drug for millions of people. And they immediately slammed into a wall. A problem that had plagued GLP-1 research from the very beginning. It's something called half-life. Half-life is simply a measure of how long a drug stays active in your body. For a drug to be effective, it needs to stick around long enough to do its job. If you take an aspirin for a headache, you expect it to work for a few hours, not a few seconds. The half-life of aspirin is about 15 to 20 minutes, but its active metabolite, salicylic acid, hangs around for hours. That's a good practical half-life. The GLP-1 your own body produces? The half-life is less than 2 minutes. 2 minutes. By the time it travels from your gut where it's released to the pancreas or the brain, it's already being torn apart. It sends its signal. Hey, food is here. And then it's gone. Vanished. This is great for a natural hormone that only needs to work for a brief moment after a meal. But for a medicine? It's a disaster. You can't ask a patient to inject themselves every 5 minutes. It's completely impractical. Now, the Gila Monsters version Exendin 4 was much better. That was the whole reason it was so powerful in the first place. Its half-life wasn't 2 minutes. It was about 2 to 4 hours. A massive improvement, but still not great. A 2-hour half-life meant that to keep a stable level of the drug in a person's system, they would need to inject it multiple times a day. The initial studies for the drug, which would eventually be named Byetta, settled on a twice-daily injection schedule. It was workable, but it was far from ideal. The holy grail of drug development is a once-a-day pill, or at the very least a once-a-day injection. Twice a day was a tough sell. The scientists at Amylin and Lilly knew they were sitting on a potential gold mine, but it was a gold mine they could only access for a few hours at a time. The question that consumed their research labs was, why? Why did these miracle molecules disappear so quickly? What was destroying them? If they could figure that out, maybe they could stop it. The culprit, it turned out, was a single ruthlessly efficient enzyme. An enzyme with the forgettable name of dipeptidyl peptidase 4. Let's just call it DPP4. DPP4 is like a molecular assassin that floats around in your bloodstream. Its sole job is to find specific peptides like GLP-1 and chop them in half, rendering them useless. It's a cleanup crew. When GLP-1 has done its job after a meal, DPP4 comes along and deactivates it, making sure the signal doesn't stay on for too long. It's a crucial part of your body's natural regulation system. But for drug developers, DPP4 was the ultimate villain. It was the gatekeeper preventing GLP-1 from becoming a viable long-term therapy. Think of it like this. You've built the most advanced high-performance race car in the world. That's your GLP-1 molecule. But as soon as it leaves the starting line, there's another car on the track, DPP4, whose only purpose is to ram into your car and wreck it within the first lap. It doesn't matter how amazing your car is if it can't even finish a single lap. The Gila Monsters Exendin 4 was like a car with slightly better armor. It could maybe last for 10 or 20 laps before DPP4 destroyed it. Better, but still not enough to win the race. Once scientists identified DPP4 as the enemy, the path forward suddenly split into two distinct strategies. It was a fork in the road of pharmaceutical innovation, and different companies began placing bets on which path would lead to success. Path number one was the direct approach. If an enzyme is the problem, then stop the enzyme. This led to the development of a whole new class of drugs called DPP4 inhibitors. These are pills like Januvia that work by blocking the DPP4 enzyme. They essentially put handcuffs on the molecular assassin. By taking a DPP4 inhibitor, you prevent your own natural GLP-1 from being broken down so quickly. You're not adding any extra GLP-1 to your system. You're just preserving the stuff you already make, letting it stick around for maybe 4 or 5 minutes instead of just 2. It was a clever strategy, and these drugs did help with blood sugar control, but the effect was modest. It was like getting your race car to last 2 laps instead of 1. An improvement for sure, but not a game changer. And crucially, these drugs didn't really cause weight loss because the boost to GLP-1 levels was just too small. This led to path number 2, the far more ambitious and ultimately far more revolutionary approach. Instead of blocking the assassin, what if you could build a better race car? A car that was invisible to the assassin, or so heavily armored that it couldn't be destroyed. What if you could re-engineer the GLP-1 molecule itself to create a version that DPP4 simply couldn't recognize or break down? A super GLP-1 that could circulate in the body not for minutes, not for hours, but for days. This was the moonshot. It required going back to the drawing board and using sophisticated bioengineering to tweak the very structure of the hormone. The goal was to keep all the beneficial parts of GLP-1, the insulin release, the glucagon suppression, the brain signaling, the stomach slowing, while changing just enough of its chemical structure to make it resistant to the destructive power of DPP-4. This was the quest that would unlock the true potential of GLP-1 therapy. It was a quest to build a molecule that was both powerful and durable. A quest that would eventually lead to the drugs we know today as Ozempic and Mounjaro. And it all started with a single brilliant idea. How do you make a key that fits the lock, but can't be broken by the janitor? The answer, it turns out, wasn't to build a better lock. It was to find a key from an entirely different universe. A key that looked and acted almost exactly like our own, but was forged in a place so harsh, so unforgiving, that it was naturally indestructible. And scientists found it in one of the last places on Earth anyone would think to look. The venom of a Gila monster. Yes, you heard that right. The breakthrough that paved the way for modern weight loss drugs came from the saliva of a venomous, slow-moving lizard that lives in the deserts of the southwestern United States and Mexico. It sounds like something out of a comic book, but it's one of the most brilliant examples of what's called bioprospecting, looking to nature to solve complex human problems. The story begins in the 1990s with a researcher named Dr. John Ung, working at a VA medical center in the Bronx. Dr. Ung was an endocrinologist fascinated by hormones, and he had a hunch that animal venoms, these complex cocktails of bioactive proteins, might contain undiscovered hormones that could be useful in medicine. It was a long shot. He spent years meticulously analyzing the venoms of snakes and lizards from around the world. Then, he got to the Gila monster. And inside its venom, he found a molecule that looked uncannily familiar. It was a peptide, a short chain of amino acids just like human GLP-1. When he sequenced it, he found it was a 53% match to our own hormone. Close enough to be a relative, but different enough to be intriguing. He named it exendin-4. And here's where it gets incredible. Dr. Ung tested what this lizard hormone did. He discovered that exendin-4 could bind to and activate the human GLP-1 receptor. Fit the lock perfectly. It sent the exact same signals. Release insulin, stop glucagon, slow the stomach, tell the brain you're full. It did everything our own GLP-1 did. But it had one crucial, almost magical property that our own hormone lacked. It was completely, utterly resistant to DP4. Remember our janitor, DP4? The enzyme that floats around and snaps the GLP-1 key in half after just a couple of minutes? Well, it couldn't touch exendin-4. The key from the Gila monster was made of a different kind of metal. To get technical for just a second, the spot where DPP4 attacks our GLP-1 is at its second amino acid, a molecule called alanine. DP4 is like a specialized pair of wire cutters designed to snip only at alanine. Exendin-4, through a quirk of evolution, has a different amino acid in that exact spot, glycine. The janitor's tool simply couldn't get a grip. It didn't fit. This was the eureka moment. Dr. Ung had found a natural, long-acting version of GLP-1. When you injected it into the body, it wasn't destroyed in 2 minutes. It circulated for hours. It could do the job of thousands of our own GLP-1 molecules, providing a sustained, powerful signal to the pancreas and the brain. This discovery was the foundation for the very first drug in this class, approved in 2005. Its generic name was exenatide, but you might know it by its brand name, Byetta. For people with type 2 diabetes, Byetta was revolutionary. Instead of their own GLP-1 signal flickering on and off uselessly, they now had a steady, strong signal that helped them control their blood sugar like never before. And a remarkable side effect was noticed almost immediately. Patients on Byetta started losing weight. It was the first real-world proof that activating this pathway for hours, not minutes, could have a profound impact on appetite and body weight. But the Gila monster's key wasn't perfect. It was a foreign substance, and it only lasted for about 6 hours. This meant patients needed to inject it twice a day, every single day. It was a huge leap forward, but it wasn't the end of the story. The Holy Grail was to create a drug that was even more convenient, even more powerful. Scientists now had a choice. They could keep searching the natural world for other alien keys, or they could try a different, even more ambitious strategy. Take our own human GLP-1 key, and figure out how to armor plate it. How to make our own, natural key immune to the janitor and resistant to being thrown out by the body. This second quest led to an entirely new way of thinking. The problem with our own GLP-1 is twofold. First, the janitor DPP4 breaks it. Second, it's a tiny molecule, so our kidneys filter it out of the bloodstream very quickly, treating it like waste. It's cleared from the body in minutes. So even if you could protect it from DPP4, it would still get flushed out by the kidneys. The challenge was to solve both problems at once. And the solution, developed by scientists at Novo Nordisk, was a masterstroke of biochemical engineering. They thought, what if we could take our tiny GLP-1 molecule and physically attach it to something enormous that already exists in our bloodstream? Something so big the kidneys can't possibly filter it out? They found the perfect anchor, a protein called albumin. Albumin is the most abundant protein in our blood plasma. Think of it as a giant cargo ship, constantly circulating throughout our body. A single albumin molecule is massive compared to tiny GLP-1. And crucially, it has a very long half-life. It sticks around in the bloodstream for about 19 days. The scientist's brilliant idea was to fuse a fatty acid chain, essentially a long greasy tail, onto the side of the human GLP-1 molecule. This fatty acid acts like a chemical tether. It has a natural affinity for albumin. So when you inject this modified GLP-1 into the body, the fatty acid tail seeks out and latches onto one of these giant albumin cargo ships. This simple-sounding modification accomplished two incredible things simultaneously. First, by hitching a ride on the massive albumin protein, the GLP-1 molecule became too big to fail, or rather too big to be filtered by the kidneys. It was no longer a tiny piece of debris to be flushed away. It was part of a massive freighter, destined to sail the bloodstream for a much, much longer time. This single change extended the hormone's half-life from 2 minutes to 13 hours. Second, this trick also solved the janitor problem. When the GLP-1 molecule was stuck to the side of the enormous albumin protein, the part of it that DPP-4 usually attacks was physically blocked. It was shielded. The janitor couldn't get his tools in there to break the key. It was like trying to fix a watch while it's bolted to the side of a battleship. This new, armor-plated human GLP-1 was a drug called liraglutide. It was first approved for diabetes under the brand name Victoza, and later at a higher dose for weight loss as Saxenda. And because it lasted for over half a day, it only needed to be injected once a day. This was a monumental improvement. We had gone from a hormone that lasts 2 minutes, to a Gila monster venom derivative that lasts a few hours, to a bioengineered human hormone that lasts all day. The science was advancing at a breathtaking pace. Each new discovery built on the last, pushing the boundaries of what was possible. They had created a once-a-day drug that effectively mimicked the body's own fullness signals, leading to significant weight loss and diabetes control. But the researchers weren't satisfied. A once-a-day injection was great, but what if they could do even better? What if they could design a molecule that only needed to be taken once a week? That quest for the ultimate convenience and power would lead to the drugs that have truly changed the world: Ozempic, Wegovy, and their even more powerful successor Mounjaro. And the science behind how they pulled that off is even more ingenious. So, how do you take a hormone that your own body destroys in less than 2 minutes and turn it into a medicine that lasts for an entire week? The answer isn't just science. It's a masterpiece of molecular engineering, a story of trial, error, and a breakthrough from one of the most unlikely places on Earth: the venom of a desert lizard. The first major problem scientists had to solve was that pesky DPP-4 enzyme, the molecular Pac-Man that gobbled up GLP-1 almost instantly. They needed to find a way to make the GLP-1 molecule invisible to it, or at least indigestible. Their search led them to the Gila monster, a sluggish but venomous lizard native to the southwestern United States and Mexico. For decades, scientists knew that the Gila monster's bite caused strange effects in its victims, including wild swings in blood sugar. An endocrinologist named Dr. John Ung, working at the Bronx Veterans Affairs Medical Center in the 1990s, became fascinated by this. He wondered what in the lizard's venom could be having such a powerful metabolic effect. After painstakingly analyzing the venom, he isolated a hormone that looked remarkably similar to human GLP-1. He named it Exendin-4. But here was the incredible part. When he tested Exendin-4, he found that it did everything human GLP-1 did. It stimulated insulin, suppressed glucagon, slowed the stomach, but it had a superpower. It was almost completely resistant to being broken down by the DPP-4 enzyme. Why? It all came down to a tiny subtle difference in its molecular structure. Think of GLP-1 as a very specific key, and the DPP-4 enzyme as a lock that is perfectly shaped to grab that key and break it. The Gila monster's Exendin-4 was like a key that was almost identical, but with one tiny bump in a different spot. That one small change meant the DP-4 lock couldn't get a grip. It just bounced right off. This single amino acid substitution was nature's brilliant solution. The lizard's hormone could circulate in the bloodstream for hours, not minutes. This discovery was monumental. It was the proof of concept that a long-lasting GLP-1-like drug was even possible. This molecule, exendin-4, was eventually synthesized in a lab and became the very first drug in this new class. Exenatide, sold under the brand name Byetta. It was a game-changer for type 2 diabetes, but it had a major drawback. While it lasted for hours instead of minutes, it still required a twice-daily injection. It was a huge leap forward, but it wasn't the ultimate goal of convenience. The next great leap came from scientists who decided to go back to the original source, the human GLP-1 molecule itself. They thought, instead of using a lizard's version, what if we could take our own natural hormone and armor it up? This led to the creation of liraglutide, the drug that would eventually be known as Victoza for diabetes and Saxenda for weight loss. And the way they did it was pure genius. They didn't just try to make it indigestible to DPP-4. They found a way to hide it in plain sight. They did this by attaching a long dangly chain of fatty acids to the side of the GLP-1 molecule. Now this might sound random, but it was a calculated and brilliant move. Here's why. Your bloodstream is full of a very common, very large protein called albumin. Its job is to transport things like hormones and fatty acids all around your body. It's like the massive, slow-moving cargo ship of your circulatory system. By attaching a fatty acid chain to the tiny GLP-1 molecule, the scientists essentially gave it a grappling hook. Once injected, this modified GLP-1 would find the nearest albumin cargo ship and latch on tight. This simple trick accomplished two incredible things at once. First, by being stuck to this enormous protein, the GLP-1 molecule was physically shielded from the DPP-4 enzymes. The Pac-Man couldn't get to it. It was like hiding behind a bodyguard. Second, and this is the most important part, it created a reservoir of the drug in the bloodstream. The vast majority of the drug molecules would be bound to albumin at any given time, just circulating harmlessly. Only a few molecules would unlatch at a time, become active, do their job at the receptor sites, and then eventually get broken down. But as soon as they were gone, another molecule would unlatch from the albumin to take its place. This extended the drug's half-life from a couple of minutes to a whopping 13 hours. They had successfully turned a hormone that lasts for 90 seconds into a medication that could be taken as a single, once-a-day injection. This was the technology that powered Saxenda, the first drug in this class to be specifically approved for weight loss. But the Holy Grail was still out there. A once-a-week injection, the ultimate in set-it-and-forget-it medication. To get there, scientists at Novo Nordisk, the company behind liraglutide, decided to combine the two strategies. They were going to create a super molecule. This molecule would become semaglutide, the active ingredient in Ozempic and Wegovy. First, they took the human GLP-1 molecule and, just like the Gila monster's venom, they made a tiny amino acid substitution in that critical spot to make it inherently more resistant to DPP-4. They fortified the lock, so to speak. But they didn't stop there. Then, they took the fatty acid chain idea from liraglutide and put it on steroids. They engineered a new, even longer and more complex fatty acid chain. A stronger grappling hook that would bind to albumin with incredible tenacity. The result was nothing short of a pharmacological miracle. This new molecule, semaglutide, was both inherently resistant to breakdown, and it anchored itself to albumin so tightly that it was released into the bloodstream at a glacial pace. Its half-life wasn't 90 seconds. It wasn't 13 hours. It was 170 hours. Almost exactly 7 days. They had done it. They had created a true once-a-week GLP-1 medication. This is the science inside the Ozempic and Wegovy pens. A molecule designed with the stealth of a Gila monster and the staying power of a cargo ship, delivering a steady, constant signal to the brain and body for an entire week, telling it, "You're full, you don't need to eat, and your blood sugar is under control." This progression from a twice-a-day lizard venom derivative to a once-a-day fatty acid-shielded human hormone to a once-a-week combination of both strategies is a masterclass in drug development. Each step built upon the last, solving a critical problem of convenience and efficacy. And for a while, semaglutide was the undisputed king of the hill, the most powerful and long-lasting incretin mimetic ever created. But even as it was taking the world by storm, another team of scientists was asking a radical new question. They realized the gut doesn't just release GLP-1 after a meal. It releases a whole cocktail of hormones. What if GLP-1 was only part of the story? What if there was another, equally powerful hormone they could target at the same time? They weren't just trying to build a better key for the GLP-1 lock anymore. They were about to build a master key that could open two different locks at once. And in doing so, they would unleash a drug with effects that made even Ozempic look tame. This new second lock was a hormone called GIP, glucose-dependent insulinotropic polypeptide. And if you think that name is a mouthful, you're not alone. For decades, GIP was the forgotten sibling in the incretin family. It was actually discovered long before GLP-1 way back in the 1970s. Scientists knew it helped release insulin after a meal, just like its more famous counterpart. But there was a problem. In people with type 2 diabetes, GIP just didn't seem to work very well. When they were given infusions of GIP, their insulin response was blunted, almost non-existent. So, for years the scientific community essentially wrote GIP off. It was considered the weaker, less effective incretin. All the research, all the money, all the pharmaceutical firepower was aimed squarely at GLP-1. It was the star player, the one that got results. GIP was left on the bench. But a few curious scientists at Eli Lilly couldn't shake a nagging question. Why would the body have two of these incretin hormones if one was basically useless? Evolution doesn't typically waste its time on things that don't work. What if everyone was looking at GIP the wrong way? What if GIP wasn't meant to be a solo artist? What if it was part of a duo? This was the paradigm-shifting idea. The theory was that the reason GIP appeared weak in people with diabetes was because the whole metabolic system was out of whack. Maybe GIP needed a healthy, functioning GLP-1 signal to do its job properly. And maybe, just maybe, if you could create a single molecule that could press both the GLP-1 button and the GIP button at the exact same time, you wouldn't just get the sum of their effects. You'd get something more. A synergy. An amplification. 1 + 1 wouldn't equal 2. It would equal 3 or 4 or even 5. This was a massive gamble. Developing a dual agonist drug is exponentially more complex than a single agonist. You have to design a molecule that can bind to two completely different receptors with just the right amount of affinity for each. It needs to be a little more active at the GIP receptor, a little less at the GLP-1 receptor, a delicate balancing act on a molecular tightrope. Get the balance wrong and you could get horrible side effects or no effect at all. It was a feat of biochemical engineering that had rarely been attempted, let alone achieved. The molecule they created was called tirzepatide. This was the master key. It was a single elegant chain of amino acids, engineered with surgical precision. When injected, it would travel through the bloodstream and, unlike semaglutide which only had eyes for the GLP-1 receptor, tirzepatide would seek out and activate both. So what does this dual activation actually do? Well, you get all the powerhouse effects of GLP-1 we've already talked about. The powerful signal to the brain saying, "You are full, stop eating." The slowing of the stomach, keeping you satisfied for longer. The signal to the pancreas to release insulin and the signal to the liver to stop dumping sugar. That's the foundation. But then you add GIP to the mix. GIP also tells the pancreas to release insulin, but it seems to do so in a more nuanced way that might be gentler on the pancreas over the long term. But the really exciting part is what it does elsewhere. There's growing evidence that GIP has direct effects on fat cells, potentially influencing how our bodies store and burn fat. It also acts on the brain but in different areas than GLP-1. So while GLP-1 is hitting the main appetite control center, GIP might be working on the reward and pleasure centers of the brain, reducing the craving and the hedonic desire for high-calorie foods. Think of it like this. GLP-1 is the powerful brake pedal slowing down your food intake. GIP is like a sophisticated traction control system, optimizing how your body uses its fuel and keeping your cravings from spinning out of control. Together, they create a level of metabolic control that is simply unprecedented. When Eli Lilly started the clinical trials for tirzepatide, first under the brand name Mounjaro for diabetes and later as Zepbound for weight loss, the results that came back were not just good. They were seismic. They sent shockwaves through the entire medical community. The trial program called SURMOUNT was aptly named because the data was literally summiting peaks no one thought were reachable with medication. With Ozempic and Wegovy, we saw average weight loss of around 15% of total body weight, which was already revolutionary. With the highest dose of tirzepatide in the SURMOUNT-1 trial, the average weight loss was a staggering 22.5%. Let me repeat that. 22.5%. That's nearly a quarter of a person's body weight. On average, many participants lost even more. These were numbers that had never ever been seen outside of bariatric surgery. A 250-pound person losing on average over 56 pounds. A 300-pound person losing 67 pounds. This wasn't just helping people lose a little weight to feel better in their clothes. This was reversing the course of obesity-related diseases on a massive scale. People were seeing their blood pressure normalize, their sleep apnea disappear, their fatty liver disease reverse, and their blood sugar levels fall from diabetic ranges back to perfectly normal. And the side effects? While still present, the nausea, the GI issues, some studies suggested they might actually be less severe than with high-dose GLP-1 alone. One theory is that GIP activation somehow counteracts some of the nausea-inducing effects of GLP-1, allowing patients to tolerate higher, more effective doses. The master key wasn't just more powerful, it might also be more elegant. The release of tirzepatide changed the game completely. It was no longer a question of if we could effectively treat obesity with medicine. The question now was, how good can we get? The success of this dual agonist approach threw open the doors to a whole new frontier of drug development. If combining two hormones was this effective, what about three? This wasn't just a new drug. It was a proof of concept. It proved that the complex, interwoven hormonal symphony that governs our metabolism could be understood and, more importantly, it could be conducted. By targeting multiple pathways at once, we could achieve results that were previously thought to be impossible. The era of the single-target drug was ending. The era of the polypharmacological, multi-pronged attack on metabolic disease had just begun. But with this incredible new power came a new set of incredibly complex problems. The world was about to find out that a miracle drug isn't a magic wand. This master key, capable of unlocking such profound biological change, was also about to unlock a firestorm of societal debate, economic strain, and deeply personal questions about the very nature of identity and willpower. The science was a triumph, but the human element was about to get very, very complicated. And so the floodgates opened. What had been a niche topic for endocrinologists and biochemists suddenly became the hottest subject on Earth. It wasn't a gradual trickle into public awareness. It was a tidal wave. Whispers in Hollywood turned into red carpet confessions. TikTok trends exploded, with users documenting their Ozempic journey to millions. Magazine covers that once preached grueling workout routines now breathlessly detailed the miracle shot that could melt fat away. The quiet, methodical, decades-long scientific pursuit was over. The era of the blockbuster weight loss drug had begun, and it was loud, chaotic, and deeply divisive. The first and most immediate complication was brutally simple: money. While the science was elegant, the price tag was staggering. In the United States, the list price for a month's supply of these medications soared past $1,000. This wasn't a one-time purchase. It was a recurring, potentially lifelong expense, akin to a second car payment or a mortgage. Suddenly, a new, stark line was drawn in society. On one side were those with the right kind of insurance. Often, coverage was only extended for a diagnosis of type 2 diabetes, or those with the disposable income to simply pay out of pocket. On the other side was everyone else. This created a bitter new form of inequality. Two people could be struggling with the exact same biological condition, the same relentless food noise, the same metabolic dysfunction. But one could afford the key to unlock their prison, while the other could only watch from the outside. The debate over healthcare in America found a powerful new focal point. Is obesity a disease to be treated, or a lifestyle choice to be managed? Insurance companies, for the most part, voted for the latter, frequently denying coverage for weight loss alone, deeming it a vanity or cosmetic issue. It was a devastating blow for millions who saw, for the first time, a real biological solution to a problem they had been told their whole lives was their own fault. The incredible demand, fueled by a media frenzy, inevitably slammed into the wall of reality. Supply. The manufacturers, despite ramping up production, simply couldn't keep up. And this led to a heart-wrenching ethical crisis. The very patients for whom the drugs were originally developed, people with type 2 diabetes who relied on them to control their blood sugar and protect their organs, were suddenly showing up to pharmacies and being told their prescription couldn't be filled. The medication they needed to manage a serious, life-threatening disease was being diverted to weight loss clinics and online pharmacies catering to anyone who could pay the price. It became a zero-sum game. One person's weight loss journey was preventing another person from getting their essential medicine. The triumph of science had created a moral quagmire. But beneath the economic and logistical chaos, a much deeper, more philosophical battle was raging. It was a battle over the very meaning of willpower. For centuries, our culture has been built on a bedrock belief that body size is a direct reflection of character. To be thin was to be disciplined, controlled, successful. To be overweight was to be lazy, gluttonous, a failure of will. This narrative was everywhere, in our movies, our advertising, our family dynamics, our own inner monologues. It was a source of immense shame and stigma for hundreds of millions of people. And then came GLP-1. This tiny molecule completely upended that entire moral framework. If a weekly injection could silence the constant, gnawing hunger, the food noise that so many had described as a relentless inner demon, then was the struggle ever truly about willpower to begin with? If the desire to overeat could be turned off like a light switch, it suggested the wiring was the problem, not the person. For many users, this was the most profound revelation of all. It was an absolution. For the first time, they felt their struggle was not a moral failing but a biological reality. The shame they had carried for decades began to lift, replaced by a quiet, righteous anger. They hadn't failed. Their bodies had. But this new perspective was met with a fierce and powerful backlash. The idea of an easy way out grated against the deeply ingrained Protestant work ethic that permeates American culture. You are supposed to earn your results through sweat, sacrifice, and suffering. Taking a shot felt like cheating, felt like skipping the hard work and going straight to the reward. Social media became a war zone. Comment sections filled with accusations. People who had lost weight through traditional diet and exercise felt their own monumental efforts were being cheapened. Fitness influencers whose entire brands were built on the gospel of calories in, calories out, saw these drugs as an existential threat. The conversation became toxic, pitting person against person, validating one lived experience by invalidating another. It forced an uncomfortable question upon society. Why do we care so much how someone else loses weight? Why does their journey have to be a hard one for us to respect it? This cultural war masked an even more intimate, personal transformation happening inside the minds of users. Losing a significant amount of weight is not just a physical process. It's a psychological earthquake. Many people had built their entire identity around being the big person. It shaped their personality, their sense of humor, how they moved through the world. When the weight disappeared, sometimes shockingly fast, it could trigger a genuine identity crisis. Who am I if I'm not this person anymore? Friends and family might say, "You look great." But inside, the user could feel like a stranger in their own skin. And then there was the silence. The sudden, shocking absence of food noise. For people who had lived their entire lives with a constant internal monologue about what to eat, when to eat, what not to eat, the guilt of eating, the craving for the next meal, the sudden quiet could be incredibly disorienting. It was like living next to a loud highway your whole life and then one day all the cars disappear. The silence is deafening. That mental energy once consumed by food was now free. But what was it free for? It forced a reckoning with other parts of their lives, other emotions and anxieties that the noise of hunger had perhaps helped to drown out. The drug didn't just change their appetite, it changed their very consciousness. It was a miracle, yes, but it was a complicated and messy one, rewriting not just bodies but minds, identities, and the very fabric of our cultural understanding of health. The battle for access and the war over willpower were just the beginning. The real questions were far more intimate, echoing in the bodies of millions of new users. What were the long-term costs of silencing your hunger? And the most terrifying question of all for those who had finally found relief: what happens when the music stops? For many, the music doesn't just stop. It's ripped away, and the silence that follows is deafening. It's a silence filled with the returning roar of a hunger that had been tamed, a food noise that had been quieted. This is the reality of GLP-1 discontinuation, and it is perhaps the most misunderstood aspect of these revolutionary drugs. The rebound is not a possibility. For most, it is a biological certainty. To understand why, we have to look past the scale and deep into the ancient survival programming hardwired into our DNA. For millennia, the human body evolved in an environment of scarcity. Losing weight was dangerous. It signaled famine, illness, a threat to survival. Your body, in its infinite and primal wisdom, developed an incredibly sophisticated system to prevent this. It's called metabolic adaptation, and it is the ghost in the machine of modern weight loss. When you are on a GLP-1 agonist, you are effectively overriding this system. You're telling your brain you're full, you're stabilizing your blood sugar, and you're allowing your body to release its stored energy. But the underlying system, the ancient programming, doesn't disappear. It's merely dormant, waiting. When you stop the medication, the external signal, the GLP-1 mimicker, vanishes. And your body doesn't just go back to normal. It panics. It perceives the rapid weight loss it just experienced not as a health victory, but as a near-fatal famine it just survived. In response, it launches a full-scale counteroffensive to regain the weight it believes is essential for your survival. This isn't a failure of willpower. This is your biology working exactly as it was designed to. The first soldier in this counterattack is ghrelin, the hunger hormone. It comes surging back, often to levels higher than before you even started the medication. Your brain is flooded with signals not just of hunger, but of ravenous primal urgency. Simultaneously, levels of satiety hormones like leptin, which have been supported by the GLP-1 drug, plummet. So you're being hit with a double whammy. A powerful drive to eat, combined with a diminished ability to feel full. The food noise returns with a vengeance, like a radio station you thought was off air, now blasting static at full volume. But it gets even more insidious. Your body also adjusts your metabolism. Your resting metabolic rate, the number of calories you burn just by being alive, slows down significantly. Your body becomes brutally efficient. It learns to run on fewer calories, hoarding every last bit of energy it can, convinced that another famine is just around the corner. This is why people who stop the medication often report that even if they eat the exact same healthy diet that helped them lose weight, they start gaining it back. Their body is now burning fewer calories to perform the same tasks. You are fighting a war against an enemy that has learned all of your strategies and has rigged the battlefield against you. This isn't theory, it's documented fact. The landmark Step 1 clinical trial for semaglutide didn't just study weight loss. It included an extension phase that followed participants after they stopped the drug. The results were a sobering prophecy. Within 1 year of stopping the weekly injections, participants on average regained 2/3 of the weight they had lost. All the cardiovascular benefits they had gained, improved blood pressure, lower cholesterol, reduced inflammation, began to reverse, tracking right along with the numbers on the scale. This single study fundamentally reframes the entire conversation around these drugs. They are not a cure for obesity. They are a treatment. And this is a critical distinction. We don't think of medication for high blood pressure as a cure. A patient doesn't take lisinopril for 6 months, get their blood pressure under control, and then declare victory and stop the medication. If they did, their blood pressure would go right back up because the underlying condition hasn't been cured, only managed. We don't expect a person with type 1 diabetes to take insulin for a year and then be done with it. We understand these are chronic conditions that require chronic management. The data from the STEP trials and others is forcing the medical community and society at large to finally view obesity through the same lens, as a chronic relapsing metabolic disease. It is not a moral failing. It is not a deficit of discipline. It is a complex interplay of genetics, hormones, and environment that creates a powerful biological drive to maintain a higher body weight, a concept known as the set point. Think of your body's weight regulation system like the thermostat in your house. For a person with obesity, that thermostat is set to a higher temperature. You can try to cool the house down by opening windows or running fans, that's diet and exercise, but the furnace, your body's biology, will keep kicking on, fighting to get back to that higher set point. GLP-1 medications are like installing a powerful new air conditioning system. They can effectively lower the temperature in the house and keep it there. But the moment you turn that air conditioner off, the original thermostat setting is still there. The furnace kicks back on with a vengeance, and the house heats right back up, often faster than it cooled down. The psychological whiplash of this experience can be devastating. Patients describe the period on the medication as a kind of freedom they've never known. Freedom from constant hunger, from obsessive thoughts about food, from the shame and self-blame. They feel like they've finally been given a fair chance, that their body is finally working with them instead of against them. To have that freedom taken away, to feel the old patterns and compulsions creeping back in, is not just disappointing. It can be traumatic. It reinforces the false narrative that they are a failure, that they lack the willpower to keep it off, when in reality, they were simply caught in the crossfire of a biological battle. They were never equipped to win on their own. This reality leads us to an uncomfortable and expensive conclusion. For the vast majority of people, the benefits of GLP-1 medications are only sustained for as long as they continue taking them. They are, for all intents and purposes, a lifelong commitment. And this raises a whole new set of monumental questions, not just for individuals, but for our entire healthcare system. The financial implications are staggering. With a price tag that can run over $1,000 a month without insurance coverage, we are talking about a commitment of well over $10,000 a year every year for the rest of a person's life. This creates a terrifying new class divide in healthcare, where a person's ability to manage a chronic disease is dictated entirely by their income or their insurance plan. What happens when someone loses their job and their insurance coverage? Do they lose their health too? Are we creating a generation of people tethered to what some have called the golden handcuffs of modern medicine, where the price of health is perpetual debt? And what about the long-term physical costs? While the safety profile for these drugs in the short to medium term is well established and generally positive, the truth is we don't have 30, 40, or 50 years of data. What does it mean for the human body to have this fundamental hormonal pathway perpetually manipulated for the majority of a lifetime? We're in the early chapters of this story, and the final pages have yet to be written. The side effects that are manageable in the short term, like nausea or constipation, take on a different character when you contemplate dealing with them for decades. This isn't a simple story of a miracle drug. It's a complex narrative about the very nature of disease, treatment, and identity. These medications have pulled back the curtain on the brute force of our own biology, proving once and for all that obesity is not a choice. But in solving one problem, they've presented us with another, far more complex dilemma. We now have a treatment that works, but it works best when taken forever. This forces a profound reevaluation, not just of the medicine itself, but of what we are willing to do, and what we are willing to pay, for a life free from the weight that has held so many down for so long. The challenge is no longer just scientific. It is now deeply personal, economic, and ethical. And at the heart of these personal, economic, and ethical challenges lies a single brutal number: the price tag. For the millions of Americans who see these drugs as a potential lifeline, the cost is staggering, often exceeding $1,300 a month. This isn't a one-time purchase. It's a financial commitment as relentless as a mortgage payment. The result is a cruel game of health insurance roulette. One person's plan might cover it with a reasonable copay, deeming their obesity a chronic disease worthy of treatment. Their neighbor with a different plan could be denied outright, told that weight loss is a lifestyle issue, a cosmetic concern not worthy of coverage. We've created a system where your access to a life-altering medical treatment depends not on your need, but on the fine print of an insurance policy you likely didn't choose. Imagine the sheer frustration. You've spent years, maybe decades, battling your own body chemistry. You've tried every diet, every exercise plan. You've endured the judgment, the unsolicited advice, the quiet shame. And then science delivers a key, a real biological tool that can finally quiet the relentless food noise in your head and rebalance the hormones that have worked against you. The key is right there, in front of you. You can see it, but it's locked inside a glass box with a 4-figure price sticker, and your insurance company is the one who decides if you can afford to break the glass. This isn't just about fairness, it's about health equity. We are witnessing the birth of a two-tiered system for managing one of the most prevalent chronic diseases in the country. A system where the wealthy can purchase a healthier, longer life, while others are left to fight the same old battles, now with the added sting of knowing a solution exists, just out of their financial reach. This economic barrier forces people into a gray market of compounding pharmacies and online sellers, a desperate search for a cheaper alternative that carries its own set of risks, with no guarantee that what they're injecting is safe, or even what it claims to be. This economic divide pours fuel on an already raging ethical fire: the issue of off-label use and the resulting shortages. While one person is fighting their insurance company for coverage to treat their type 2 diabetes or life-threatening obesity, another, with a much lower BMI and a desire to lose 10 pounds before a vacation, can walk into a Medi-Spa and get a prescription with cash in hand. Hollywood and social media influencers flaunt their rapid, effortless-looking transformations, rebranding a potent medical treatment as a luxury wellness product. This explosive demand from the vanity market has created critical shortages across the nation. Pharmacists have to tell frantic parents they can't fill their child's diabetes prescription. Patients who have been stable on these drugs for months, finally feeling in control of their health, are suddenly unable to get their medication, risking a rebound in weight and a dangerous spike in their blood sugar. This forces us to ask an uncomfortable question: who deserves this medicine? Is it the person with a BMI of 45 and a host of comorbidities? Or the person with a BMI of 28 who is deeply unhappy with their body and willing to pay out of pocket? Is a doctor who prescribes Ozempic for cosmetic weight loss acting unethically if they know it contributes to a shortage for diabetics? There are no easy answers. The situation highlights a profound tension in our society. We simultaneously stigmatize obesity as a moral failing, yet we also judge those who use a medical tool to treat it, calling it cheating or the easy way out. We celebrate thinness as the ideal, creating immense pressure to conform, and then we're shocked when a new tool to achieve that ideal is used by those who aren't sick enough. Perhaps the most challenging ethical question is whether these drugs are truly solving the problem, or simply medicalizing our cultural obsession with thinness. Instead of fostering a world of body acceptance and focusing on health at every size, are we just offering an incredibly expensive lifelong subscription to an idealized body type? And for those who do get access, for whom the cost is not a barrier and the prescription is medically sound, the journey is profoundly personal. It is anything but the easy way out. The side effects, the nausea, the fatigue, the digestive upheaval can be grueling. But for many, the benefits transcend the numbers on a scale. It's the quiet miracle of sitting down to a meal and feeling satisfied with a few bites, instead of being consumed by a craving that feels like a separate entity living inside your brain. It's the newfound energy to climb a flight of stairs without losing your breath, or to get on the floor and play with your children without your joints aching in protest. It's the simple dignity of fitting comfortably in an airplane seat, or shopping for clothes in any store you choose. These are not small victories. They are the reclamation of a life that felt like it was shrinking, day by day. It's the reduction of food noise, a concept that people who haven't experienced it struggle to grasp. Imagine a constant radio in your head, always broadcasting thoughts about your next meal, what you shouldn't have eaten, what you desperately want to eat. GLP-1 agonists for many finally turn down the volume on that radio, creating a mental peace they haven't known in decades. Yet, this new life comes with a golden handcuff: the understanding that this is likely a lifelong commitment. This is not an antibiotic you take for 10 days to cure an infection. Obesity is a chronic disease, and these drugs are a management tool. Stop the medication, and the biological signals it was suppressing come roaring back. The appetite returns, the food noise gets louder, and in most cases the weight comes back. This reality is a critical, often overlooked part of the conversation. It means committing to weekly injections indefinitely. It means a perpetual line item in your budget, and a constant reliance on the pharmaceutical and insurance industries. And it means accepting the unknown. While the short-term data is incredibly promising, we simply don't have 30 or 40 years of data on what it means for a human body to be on these powerful metabolic drugs for the majority of a lifetime. We are, in essence, the first generation of a global real-world experiment. But the science isn't standing still. Even as we grapple with the implications of Ozempic and Mounjaro, the next wave is already on the horizon. Researchers are developing triple agonist drugs that target GLP-1, GIP, and another hormone, glucagon, promising even greater weight loss and metabolic benefits. There are oral versions in late-stage trials that could eliminate the need for injections altogether. The pharmaceutical pipeline is brimming with new compounds designed to be more potent, have fewer side effects, and perhaps even target the Holy Grail: preserving muscle mass during weight loss. We are at the very beginning of a therapeutic revolution. The Gila monster's venom didn't just give us a new drug. It unlocked a whole new doorway in our understanding of human metabolism. So where does that leave us? We've journeyed from the saliva of a desert lizard to the frontiers of biotechnology, from the intricate dance of hormones in our gut to the complex calculus of healthcare economics and ethics. We have seen how a single molecule can quiet a lifetime of food noise, reverse chronic disease, and spark a cultural phenomenon that is reshaping our society. These medications are not a magic bullet. They are not a substitute for healthy habits, nor are they a cure for the societal pressures that warp our relationship with our bodies. They are a tool, a profoundly powerful, biologically complex, and socially disruptive tool. They have exposed the deep fault lines in our healthcare system, our cultural biases around weight, and the desperate yearning for a solution to a problem that is both deeply personal and globally shared. The science has given us a new and powerful answer to the question of how to regulate appetite, how to control blood sugar, how to lose weight. But it has left us with a set of far more difficult questions. Who gets access to this power? What is the true price we are willing to pay for it? And as we continue to unlock the secrets of our own biology, what kind of future are we building for ourselves? The conversation and the science is only just beginning.